Hello, everyone. I'm really delighted to have this opportunity to talk to you today about our research. And I will talk about the pathophysiology of muscle in diabetes. And the outline for my talk is to, first of all, overall, I'm going to talk about the effects of physical activity and diet on metabolism. And one of the concepts that we put forward is that exercise is a medicine. And we're very interested in rhythms, and we're interested in circadian rhythms. So that's a big part of my presentation today. And I'm going to start my talk by giving some of the basics around circadian biology and how that affects metabolism. And then in the second part of the talk, I'm going to really share with you some ideas about how we can synchronize exercise and diet interventions with this molecular circadian machinery to enhance insulin sensitivity, and how this might be beneficial for people with obesity or diabetes. So what we really appreciate today is that within nearly every cell of our body, we have an internal clock. And this circadian clock anticipates and adapts our physiology to different phases of the day. And so you can imagine that there's certain times of the day where we have better coordination, faster reaction times, certain times of the day where body temperature or blood pressure may be higher or lower, depending upon that clock. And some of this biology may be related to a circadian or diurnal change in hormone levels, like melatonin or stress hormones, cortisol, or insulin. So our biology is really steered by this biological clock, and it helps us to regulate sleep patterns, feeding behavior, hormone release, etc. And so we're really interested in this clock, and we're interested in rhythms. We're interested in daily rhythms in metabolism, rhythms in free fatty acid availability, insulin sensitivity, even oxidative capacity. Now, I mentioned earlier that nearly every cell in our body has a circadian clock. And this clock is controlled by a transcription translation feedback loop. This is a gene regulatory network, a transcription translation feedback loop that controls the expression of thousands of genes throughout the day in multiple organs. So there's a clock in muscle, a clock in fat, a clock in liver, and all of these clocks are synchronized by the central clock in the suprachiasmatic nuclei in the hypothalamus. So we're interested in how these clocks can steer metabolism in a time-dependent manner. And we're interested in how exercise might be a Zeitgeber, a timekeeper, to help reset this clock. Now, here's an example of how every cell in the body has a clock. And we approached this question by studying primary human muscle cells, and we were able to synchronize these cells with serum shock. And you can see on the right that we're looking at different gene profiles in these muscle cells that are synchronized with a Zeitgeber, serum shock. And we can see rhythms in the expressions of rhythms in the expression of genes. We can see rhythms in the epigenetic signatures in muscle. And these rhythms in these gene patterns and these epigenetic signatures are related to genes that are controlling metabolism or developmental processes. Now, if we look within skeletal muscle from people who have obesity, we can see that with obesity, there's a disruption in some of these gene expression of the clock genes. So in men and women who have obesity, we can see that with obesity, there's a disruption of the expression of genes that are central for the clock, shown in the upper right-hand panel. And that if individuals go on a weight loss therapy, or if they have gastric bypass surgery and they restore their body weight, the weight loss can upregulate or control the expression of these clock genes. So we can see that obesity is associated with the disrupted clock, and weight loss can improve this clock machinery. Now, if we look at some of the correlates between the expression of these clock genes, we can see that one of the factors that correlates with the expression of clock genes is plasma lipids. And we can see that in individuals who have a higher elevation in plasma lipids, they have a greater disruption of the clock machinery. And if we take muscle cells from normal glucose tolerant people and if we culture them over time, we can see that there's a rhythm in the expression of many genes. And if we expose these cells to high levels of lipids, the lipid exposure disrupts the clock expression in human muscle cells. So what does this mean? This suggests that people with obesity have disrupted skeletal muscle clock gene expression, that the plasma lipid levels are correlating with the clock gene expression, suggesting that disruption of metabolic homeostasis can somehow modify or downregulate the clock gene expression. And that if we incubate cells in culture, we can directly disrupt the clock by exposing these cells to lipids. So there's an influence of obesity, weight loss, and lipid levels on the muscle clock. Now, we were really interested in this clock gene machinery, and we asked whether or not there was alterations in clock gene machinery and clock gene function in muscle from people who have diabetes. And so we took muscle biopsies from people with normal glucose tolerance or type 2 diabetes. We cultured them over time, synchronized them so we could align all the clocks in every fiber. And we did a gene expression analysis, and we also looked at oxidative capacity with a seahorse analysis. And what this shows in the blue graph on the bottom is that we have a rhythm in our oxidative consumption. So individuals with normal glucose tolerance have a rhythm in their metabolism. And this is very similar to what Patrick Schrawn and his team has seen in muscle biopsies that are taken from people who have normal glucose tolerance. So the question we had was whether or not there was a rhythm in muscle cells from people who have diabetes. And in fact, what we found was that in myotube cultures taken from people with type 2 diabetes, there was a disruption in the rhythm of the gene expression over time. And you can see on the right, this is kind of a cartoon of our findings. You can see that there's a very dynamic change in the expression of genes throughout the day in healthy individuals. But in muscle taken from people with diabetes, there is metabolic inflexibility, and there is an inflexibility in the diurnal gene expression profile. And this may be one of these signatures of the insulin resistance in people with diabetes, a disruption in the rhythms of the gene expression. So the other thing we were interested in was whether or not the changes in the gene expression had an effect on any metabolic outcome in these muscle cells. And over the years, we've been studying these primary muscle cultures from people with normal glucose tolerance or type 2 diabetes. And we've been able to show in the right-hand panel that insulin promotes a robust increase in glucose uptake in muscle cells from the healthy individuals. But in muscle cells from the people with diabetes, there's a strong insulin resistance. So we took our myotube cultures and we measured oxygen consumption in these myotubes, and we could see that in blue, there was a rhythm in the oxygen consumption rate in the myotube cultures from the people with normal glucose tolerance. But in red, you can see that the rate of oxygen consumption is dampened, and there's a metabolic inflexibility in the rate of oxygen consumption in the myotube cultures from these people with type 2 diabetes. So what's interesting here is that these cultures maintain an insulin-resistant phenotype from the donor, from the type 2 diabetic donor, even after several passages. So we were interested in the mechanism by which these clock genes were disrupted and how the disruption of the clock can impair metabolic function. So what we did was we did a gene ontology analysis shown in the upper left-hand corner, and we found that in muscle taken from people with diabetes, there is a disturbance in mitochondrial function in the gene expression profile of the mitochondrial matrix and genes that are annotated towards mitochondrial function. And the other thing we could show is that if we take one gene, OPA1, which is a gene that regulates inner mitochondrial membrane morphology and function, if we study that gene in culture, and if we knock it down in muscle from mice, we can show that OPA1 can disrupt mitochondrial function, and that also disrupts the clock gene expression. So this would suggest that there's a link between mitochondria function and clock function. So somehow these two processes are interconnected. And if we then take myotube cultures from people with normal glucose tolerance, and we use siRNA to silence the expression of this mitochondrial gene, OPA1, we can show that silencing OPA1 expression, knocking that out of muscle, also impairs clock gene function in myotubes. So what we have is a relationship between the mitochondrial function, the clock function, and the manifestation of insulin resistance with obesity or diabetes. So what our evidence suggests is that there is a bidirectional relationship between the mitochondrial function and the clock gene function. And that in cases of insulin resistance, obesity, or type 2 diabetes, there's a dampening of the oscillations of the clock gene expression over time. And this is somehow also coupled to the mitochondrial dysfunction that we see. And so that there is a bidirectional relationship between these two processes. So what we find is that there is a reduction in the amplitude of the clock genes. This clock gene amplitudes are depressed in type 2 diabetes. We also find that in myotube cultures from people with type 2 diabetes, there is a disruption of the mitochondrial rhythms. We also find that at the molecular level, the clock proteins are binding to the intermitochondrial membrane promoters. And this suggests that somehow the clock is steering the expression of these mitochondrial proteins in a diurnal fashion. And that if we disrupt the intermitochondrial membrane, we can also change the clock rhythms. And so there's a bidirectional relationship between clock on one hand and mitochondrial function on the other. All right, so with that as some background, we can already have evidence that there is a disturbance in the circadian rhythms in muscle from people with diabetes, and that this disruption is associated with an alteration in mitochondrial function. So we wanted to ask whether or not we could reset some of the rhythms in muscle from people with diabetes by using either exercise or diet. And we would like to use exercise or diet as a way to reset the clocks and restore metabolism. Now, diet is interesting because there is evidence to suggest that if you modify the time of day that you're eating, that may also have a positive effect on metabolic outcomes. In other words, people who are using a time-restricted feeding paradigm, they have a prolonged fast. So there's a greater number of hours that they're fasting. And that essentially gives the body sort of a break. And it allows the body to have a state of a prolonged fast. And this prolonged fasting is associated with improvements in whole body insulin sensitivity, improvements in glucose and lipid parameters, improvements in blood pressure, etc. And so there's many more studies that have been performed in mouse models where mice have been given long periods of feeding or shortened periods of feeding. But we wanted to take a study in humans. We wanted to ask this question in humans. Would we be able to study the effects of timing of food on metabolic outcomes? And so for this study, we performed a crossover design. The study was performed in men with obesity. The men came into our laboratories and they either had an extended feeding paradigm in black, where they were given food between 7 o'clock in the morning and 9 o'clock at night, or in red, they had a time-restricted feeding protocol where they were allowed to have meals between 10 o'clock in the morning and 5 o'clock in the afternoon. And the diet was kept the same between these two groups. The amount of calories that they ate was the same. The composition of the diet was the same. The only difference was the window in which they were allowed to eat food, extended feeding paradigm or time-restricted feeding paradigm. And what we did was these men came into the lab. They underwent either the extended feeding paradigm or the time-restricted feeding paradigm for 5 days. On the last day, we took muscle biopsies and blood samples every 4 hours across a 24-hour period. And we took these samples and we measured gene expression and metabolite abundance in these samples. And so what we found here, first of all, was the gene expression changes shown on the left. And we always have the extended feeding paradigm in black. The time-restricted feeding paradigm is in red. And first of all, what we found was that this 5-day of time-restricted feeding did not alter the rhythm of the clock gene expression. And this is important because the clock is evolutionarily conserved. And so it suggests that just changing your feeding window is not sufficient to be able to disrupt the clock or improve the clock. But what we found was the way the clock was steering the expression of genes was altered. And we found, shown on the right, the patterns of gene expression and the patterns of metabolites, the way in which the muscle utilized substrates, was changed. We found that when these individuals had their time-restricted feeding paradigm, there was a change in the rhythmicity of amino acid transporters and amino acid metabolites. And this may be because the men were having a prolonged fast, and there was a greater reliance on amino acids as substrates to overcome this fasting period. So you can see on the left are some different gene ontology pathways that were different between the extended feeding and the time-restricted feeding paradigm. And they were mainly related to transporter activity. And shown on the right are waffle plots for the metabolite profiles. And we can see that with the time-restricted feeding paradigm, this is always the second set of bars for each time period, there's a greater shift in these metabolites to be more amino acids and more lipids. And so there's a greater reliance on lipids and amino acids as fuel sources. So what we have is some evidence that time-restricted feeding can alter the lipid and amino acid metabolite rhythmicity, but that is not because of a change in the clock gene machinery. The expression of the core clock genes is unaffected by this short-term feeding paradigm, but there is a change in the way in which the muscle cells are performing metabolism. There's increased amino acid metabolite rhythmicity, and the time-restricted feeding induces rhythmicity in a subset of transporters for amino acids. All right, so I've provided some evidence that in people with obesity or diabetes, there's a disruption in the clock gene function. And I've also shown that there's, over the course of a day, there's rhythmicity in the way in which muscle cells perform metabolism. And so we're really interested in whether exercise can be a way in which we can, steer metabolism. And so this is an important question for us, because if you look on the left-hand panel, you can look at continuous blood glucose monitoring data from people who have normal glucose tolerance in the lower curve and people who have type 2 diabetes in the upper curve. And you can clearly see that when people have type 2 diabetes, there's a great dysregulation of glycemia. These individuals have extreme hyperglycemia in conjunction with meals. And we were very interested in whether or not exercise, either in the morning or in the afternoon, would dampen some of these glucose excursions that are characteristic of people with type 2 diabetes. And so what we did was we had men with diabetes come into the laboratory, and they performed a crossover study. And they were introduced to high-intensity interval exercise training. These men performed exercise either in the morning or in the afternoon for 2 weeks. And then they had a 2-week washout period. They came back in, and then they did the exercise at the opposite time of the day. And so what we found was that when the men performed exercise in the afternoon, and this is shown in the blue curve, compared to their baseline in gray, we found that when the men performed afternoon exercise, there was an improvement in blood glucose control, both at the first week and the second week of training. However, when the same men performed exercise in the morning, they actually, shown in red, they had a worsening of their blood glucose control. And you can see that after the first week of training and after the second week of training. So the evidence suggests that the afternoon exercise had an improvement in blood glucose control in these men with type 2 diabetes, but the morning exercise had a worsening of their blood glucose control, which was surprising and unexpected. So then we actually went and took muscle biopsies and fat biopsies and serum samples from these men. And we asked whether or not the time of day of exercise affected the way in which muscle and fat performed metabolism. We first looked at the muscle proteome. We surveyed all the proteins expressed in muscle, and we found that irrespective of the time of day of exercise, there was a training effect on regulating the abundance of proteins in muscle. However, we also found that there was a time of day dependent effect of exercise on the muscle proteome. Now, if you look in the red bars down on the bottom, what we found was that the morning training was associated with an alteration in the abundance of proteins that were associated with glucose metabolism. And if you look in the blue, the afternoon training was associated with an alteration in the abundance of proteins that was associated with lipid metabolism. So what this suggests is that the morning training provoked glucose metabolism, and the afternoon training provoked lipid metabolism. Now, I mentioned that we did a metabolomic analysis on muscle and fat and also serum. And we looked at the way in which these organs did perform metabolism. And what we found was that irrespective of the time of day of exercise, there were many metabolites that were changed with training. And some of these metabolites were related to diacylglycerol metabolism, energy metabolism, lipid metabolism, acetylcarnitines, etc. But we also found a time of day dependent effect of exercise on the metabolome, with morning exercise primarily affecting carbohydrate metabolites in the pentose phosphate shunt, and afternoon exercise primarily affecting lipid metabolites in and also proteins that were associated with the mitochondrial complexes. So this suggests that there is a time of day dependent effect of exercise on the way in which muscle and fat cells perform metabolism. So the lab is really interested in this question, and we think that some future directions and questions that we're interested in are whether what are the physiological and molecular events that promote these time of day dependent effects of exercise on muscle and fat and serum metabolites, and also muscle and fat protein abundance. We're also interested in whether or not the time of day of exercise differences we see are related to the diet that the people ate or other behavioral changes. And we're also interested in whether or not the differences we see are dependent upon the sex of the individual. But we have some pilot data now where we've really controlled for the diet, and we're really giving these people calibrated meals at very timed intervals. And we find that even when we control for diet in a careful way, we find that the morning exercise is still associated with an elevation in the plasma glucose, but the afternoon exercise is associated with an improvement in blood glucose controls. And now, of course, this is a special kind of exercise. It's high intensity exercise, but it provides evidence to suggest that there's effects of exercise timing on metabolism. Now, our work is not the only work to suggest this. Currently, there's several research teams around the world have shown that afternoon exercise is more efficacious than morning training for metabolic outcomes. There's the data I showed you for the improvements in blood glucose control with afternoon exercise, but there is a Dutch group that's shown that in whole body insulin sensitivity is improved after afternoon exercise. There's a Norwegian group that has shown that afternoon exercise improves blood glucose control and nocturnal blood glucose measured by continuous blood glucose monitors. There are two groups that are related to large data sets with the UK biobanks. One shows that midday afternoon exercise is associated with lower risks of all-cause mortality, morbidity, and cardiovascular mortality. And another paper that just came out this year showing that moderate to vigorous physical activity improved glycemic control in adults with diabetes. So evidence is emerging that there may be time of day dependent effects of exercise on metabolism in people with diabetes. And some of this can be related to diet as well. Okay, I won't show you all the data from this paper, but we've been able to dissect some of the mechanisms for this time of day dependent effect of exercise on metabolism in mouse models. And we've been able to study mice during the early rest phase, essentially when mice should be sleeping, or the early active phase. And this is essentially 3 hours into the wake period for a mouse. And we've shown that this active phase, this early morning exercise in a mouse, is associated with improvements in many of the metabolic outcomes in a healthy mouse on glycolysis, lipid oxidation, and ketone metabolism. So timing of exercise can matter. All right, so some of the gaps in the field are really to try to, look at muscle. Many of these omic studies that we've been doing are limited to muscle. Several organs are important for the control of metabolism, and the field is really getting a greater appreciation for how many different organs come together to orchestrate metabolism. And so this idea of organ crosstalk is important. And the next part of my talk will consider how many organs in the body communicate to steer metabolism. And I'll show you a few examples. And one of the mediators of this crosstalk has been something identified over 60 years ago as something called the Goldstein factor. And now we understand that these factors are called exerkines. They're factors that are secreted from the working muscle and other organs from animals in response to exercise. This is a work that was just published this year by Jonathan Long, and it's a beautiful study showing how different organs can secrete factors into circulation to start to provide communication from one organ to the next, to help the body control systemic homeostasis. And one of the factors identified by Jonathan Long is a factor called human carboxylesterase 2. It's called CES2. And our team was able to study this exercise responsive factor, CES2, on metabolism. And we could show that this exercise responsive factor, CES2, when administered to animal models or cells, was able to improve hepatic steatosis. So that's just some examples of how these exercise factors can improve some of the metabolic disturbances seen in people with obesity or diabetes. And this is sort of a summary of what we know in the field today. Exercise has a powerful effect on multiple organs, not only muscle, also on the islets, the beta cells, the fat cells, the heart, the liver, and many factors are released from these organs to help communicate information about the metabolic state of each organ to help the whole body adapt to the training. Many of these factors are proteins, but the field is starting to appreciate that metabolites can also signal information about the metabolic status of multiple organs in the body. And one of the first exerkines identified was lactate, and that's a metabolite that's released in response to exercise. So our team has been trying to survey multiple organs in the body, muscle, liver, heart, the hypothalamus, different fat tissues, and we've been looking at the metabolomic profiles of each of these organs, and we've been trying to see how these metabolite profiles between each of these organs are correlated in response to exercise. So if you look on the left, you see this circus plot of metabolites in a sedentary animal. And on the right, you see this circus plot of the metabolite correlations between organs in an animal who is exercised. And you can see this great disruption in metabolic homeostasis with exercise. And we've been able to show that many of the metabolites that change with exercise, they're related to epigenomic pathways. And there's greater changes in these metabolites when the animals perform work during the early active period. So there's a time of day dependent effect of exercise on these metabolite profiles. And one of the metabolites that we could characterize, a new exerkine, is something called 2-hydroxybutyrate. And I won't go through all the data, but this metabolite is upregulated in terms of early active exercise. And if we administer this metabolite to an animal, it shifts substrate utilization, and the animal oxidizes more fat and it spares glycogen and glucose. So it's an example of a novel exerkine that is released in a time of day dependent fashion. So I can just sort of sum up. So what we're interested in is this metabolic crosstalk, the way in which different organs communicate information about stress. Exercise is a stress. we can see that there's a time of day dependent effect of exercise on the way in which muscle and liver communicate information. Looking at the left versus the right, you can see that exercise during the early active phase has a greater disruption of metabolic homeostasis. Some of this may be related to whether an animal is fasted or fed. If you look at the left, when an animal is exercising during the early rest phase, they're mainly fed. They have a lot of nutrients on board. When they exercise, they have substrates on board in the muscle, in the fat, in the liver to help the animal cope with this metabolic stress. If you look on the right, when the animal exercises during the early active phase, they're coming off an overnight fast. They really have lower levels of glycogen. They're mainly utilizing lipids as a substrate. When the exercise is performed, it's a great metabolic stress to the animals, and they have a massive alteration in the metabolite levels. All right, so the team now is trying to understand this circadian control on metabolism. I've provided evidence that there's changes in the clock gene expression and metabolic genes with obesity or diabetes. We're interested in exercise and how that can reset the metabolism. We think it may involve some of these energy sensors like AMPK or nutrient sensors like mTOR or an oxygen sensor like HIF1 alpha. And these sensors can interact with the clock machinery, and it can modify mitochondrial function and substrate utilization. So just wrapping up, the conclusion is that exercise training enhances insulin sensitivity. It improves blood glucose control, even in people with diabetes. We can find that time-restricted feeding alters lipid and amino acid metabolite rhythmicity without perturbing the clock gene machinery. And I've shown you that time of day of exercise can specify the rewiring of metabolic pathways and systemic energy homeostasis. So exercise is good, but timing the exercise might amplify the response of the training. So with that, I want to thank you for your attention. And again, it was a pleasure to be able to share our work with you. Thank you.